Hi everyone, welcome back to my educational channel on biology. In this video, we are going to cover the last part of subtopic 15.2 of the Form 5 Biology KSSM syllabus. The title is Comparison between Spermatogenesis and Oogenesis. Our learning standards are that at the end of this video, we should be able to compare and contrast between spermatogenesis, as shown here, which is the formation of sperms in the testes, and oogenesis, which is the formation of the ovum or the secondary oocyte in the ovary. So we are going to discuss the similarities and the differences between spermatogenesis and oogenesis. So here is a side-to-side comparison of spermatogenesis and oogenesis. Now basically for us to memorize easily, we have to see the similarities, okay, between spermatogenesis and oogenesis. They are quite similar in certain aspects. For example, in spermatogenesis, the process starts off with mitosis, then development, and then meiosis, and then differentiation. Now in oogenesis, there's first of all mitosis, and then development, growth and development, then meiosis 1 and meiosis 2, but there's no differentiation here, right, of the ovum. Apart from that, both of these processes start off with the primordial germ cell, which is diploid. Okay, primordial germ cell that's diploid. Then also take note that the ending, the word, okay, the syllable at the end sounds the same, gonium. For example, in spermatogenesis, primordial germ cells divide by mitosis to form spermatogonium. But for oogenesis, primordial germ cells undergo mitosis to form oogonium. So the spermato here, okay, is replaced by oo, right? So primary spermatocyte for spermatogenesis, but in oogenesis we have the primary oocyte. The site remains the same, CYTE. Okay, right. So let's do a more thorough comparison for the exam. Okay, for exam questions, like essay questions, they may ask you what is the difference between spermatogenesis and oogenesis. Okay, so let's look at the next slide. Now let's answer this question. Compare and contrast between spermatogenesis and oogenesis. 10 marks. So this question can be asked in the essay section, and it can also come out as a structured question in the SPM or in the exams. Now, here we have 2 similarities and 8 differences. So each similarity or each difference is 1 mark. Now, number 1, what are the similarities? So there are 2 similarities. Both spermatogenesis and oogenesis are processes of gametogenesis. That means processes whereby gametes are produced that take place in the reproductive organs, namely the testes and the ovaries. Now, both spermatogenesis and oogenesis produce haploid gametes, meaning the sperm and the ovum, which are involved in fertilization. So in fertilization, the nucleus of a sperm will fuse with the nucleus of an ovum in order to produce a diploid zygote. Okay, so both the gametes are involved in fertilization. Now, the differences from the aspect of the site or the location where the process occurs. Spermatogenesis takes place in the testes of males, but oogenesis takes place in the ovaries of females. Secondly, what are the products formed after meiosis 1? So after meiosis 1, in males, 2 secondary spermatocytes are produced when one primary spermatocyte divides. Okay, so 2 secondary spermatocytes are produced from one primary spermatocyte. But in oogenesis, after meiosis 1, there are less functional cells produced. Only one functional secondary oocyte is produced, and another cell produced is a smaller non-functional polar body. Okay, so all these are produced from one primary oocyte. Now, how is meiosis completed? In spermatogenesis, meiosis 1 and 2 is completed in a continuous process without stopping. Now, for oogenesis, meiosis 2 is only completed if and when a sperm fertilizes the secondary oocyte, when a sperm penetrates the secondary oocyte. So if there's no fertilization or the sperm does not penetrate the secondary oocyte, meiosis is not completed at all. Okay, and the secondary oocyte will remain a secondary oocyte. It will not undergo or complete meiosis 2 to become an ovum at all. Okay, if there's no fertilization. So next, difference number 4. Does differentiation occur? In spermatogenesis, the spermatids formed after meiosis 2 do undergo differentiation. That means changes in the shape and structure to become sperms. So sperms will have the head, midpiece, and tail after the spermatids have undergone differentiation, in order to help them carry out their function efficiently, that is to swim towards the ovum for fertilization. Okay, that's why the spermatids undergo differentiation to become sperms. But in oogenesis, the secondary oocytes formed after meiosis 1 and the ovum formed after meiosis 2 do not undergo any differentiation. So the ovum, that's the final product of oogenesis, is just a round cell. Okay. Now, what is the final product of meiosis for spermatogenesis and oogenesis? So, in spermatogenesis, one diploid spermatogonium produces 4 haploid sperms after meiosis. Okay, we're talking about the number of daughter cells produced. Okay, 4 haploid sperms are produced after meiosis. So, recalling back the process of spermatogenesis, actually from one spermatogonium, right? The spermatogonium will develop to form one primary spermatocyte that is diploid, and then this primary spermatocyte will undergo meiosis 1 and 2 and finally produce 4 spermatids that differentiate to form 4 sperms. So from one parent cell, which is the spermatogonium or the primary spermatocyte, 4 sperms that are functional are produced at the end of the spermatogenesis process. But in oogenesis, one diploid oogonium produces only 1 functional haploid ovum and 3 non-functional polar bodies after meiosis. Again, let's recall oogenesis. In oogenesis, one oogonium that is diploid develops and grows to become one primary oocyte that is diploid, which undergoes meiosis 1 and 2 to form 1 ovum and 3 polar bodies, which are all haploid. Okay, so here we see that one parent cell, which is the oogonium or the primary oocyte, produces only 1 functional cell, which is the ovum, and the other 3 are non-functional polar bodies after the process of meiosis. Now, what is the structure of the gametes? Sperms are smaller than the ovum or the secondary oocytes, and their structure is such: they are made up of the head, midpiece, and tail region. Okay, whereas the secondary oocytes, which are the immature ovum or ova, are larger, much larger. They are the biggest cells in the body, and they are spherical in shape. Next, let's discuss the amount of gametes produced through spermatogenesis and oogenesis. In spermatogenesis, millions of sperms are produced every day in a male. Okay, so that's quite productive. But in oogenesis, the process is less productive in the female because only one secondary oocyte, which is the immature ovum, develops and is released from the ovary of a female in each menstrual cycle that lasts 28 days. That means every 28 days, only one or a few more secondary oocytes develop and are released. Okay, usually it's only one secondary oocyte. Now, the production of gametes, is it continuous? And the age when gametogenesis starts and ends. So in spermatogenesis, the production of sperms is continuous, starting from puberty, when the male is 14 years old, and it continues until old age. So, but for oogenesis, the production of the secondary oocyte is not continuous. So oogenesis actually starts in the female fetus, and then when primary oocytes are formed, they will remain dormant when the baby is born. That means they are inactive once the baby is born, and until 12 years later when the female reaches puberty. Then the process continues, okay, and the primary oocytes become active again. So they will undergo meiosis 1 to form the secondary oocyte. And this is only a few at a time. So oogenesis will stop during menopause, that means from 50 years onwards, around 50 years. Right, that's all for this lesson. So please share, like, and subscribe to my channel if this lesson has benefited you. Thanks, and see you in the next video.